Good morning everyone. I hope you can hear me. Welcome to this morning's sessions. I hope you recovered from last night conference dinner. It was really great. So we are in the plant pathogen interactions in multi-stress conditions session. We have quite a few different talks on viruses, fungi and bacteria. So myself and Manuella were invited speakers, and also we asked to be a chairs. So what we decided to do to share this time slot, because we got quite a few abstracts, and to just give more opportunities to other people to present. So I'm going to start the session with short overview on plant vector virus interactions, and then Manuella will do a talk on viruses and water stress. So I'm Piotr TREBICKI. I'm from Macquarie University and also associated with University of Melbourne in Australia. I'm going to give a bit of an overview on plant virus insect interactions, mainly under elevated CO2 as part of the climate change, but also about the temperature. So biotic stresses are quite harmful for the plant production, and when you have like combined stresses, that's even more so. There's quite a few different ones that we can highlight. I guess temperature, drought are the most important ones, and in a context of climate change, we are expecting that they're going to intensify and going to increase. So given that we need to be quite vigilant about food projections and whatnot, it's sort of an important thing to consider. So this slide I don't think requires much explanation. Just want to mention that most of the CO2 emissions happened in the last few decades, and they're going to double by the end of the century when we look at those pre-industrial revolution levels. And obviously, following that, temperature is going to increase. This is the 100-year average temperature, and as you can see, for last few years, we are about almost 1 degree higher than the long-term average. And predictions or modeling suggest that given current emissions or lots of emission scenarios, we are looking at 2 to 4 degrees higher temperature to what it used to be 100 years ago. So obviously, elevated CO2 causes, warming and such, and because of warming, we have more like energy in the system, much more violent sort of weather events, like extreme weather events, less available land for production. And I think the main thing is this unpredictability, where we can and when we can grow food. The other thing is that we're looking at population increase, and to meet the demand of population increase, we have to double food production, which is a bit of a challenging. I'm just showing here a map of Australia where we actually can grow food. So, for a big continent like in Australia, we don't have that much space, and predictions are that this area is going to shrink. So I think it's very critical to understand insect-plant pathogen interactions. Obviously, disease are sort of function of those interactions, and different parts of this disease triangle, as you can see, going to be affected in different ways. So we have to be smart about it. We have to understand how temperature, how elevated CO2, how are the factors that are part of the climate change going to affect different parts of this, and what kind of scenarios or what kind of situation is going to be conducive for increased crop loss. And understanding that, we can sort of adapt, or we can anticipate and work with the crops to be more resilient. So just very briefly, elevated CO2 or CO2, it's good for plants because it's like fertilization. Obviously, plants need the CO2. So in general, in wheat, for example, we have, when we're just looking at the CO2 itself, we have around 22% yield increase. We have greater water use efficiency, and we have increased surface area. But on the flip side, we have big effect on nutritional quality, like micro macronutrients, and grain, so basic grain protein reduction. But when we add temperature into the mix, obviously the whole situation doesn't look that great. And the big problem is across the board with elevated CO2, that the quality of the food produce are decreasing. So we've done research when we grew wheat under elevated ambient CO2, and there was lots of dough rheology. As you can see here, there's less quality bread and elevated CO2 in terms of volume and macro micronutrients and glutens. When it comes to insects, in my case, of my interest, insect vectors, direct effect of CO2 is negligent, so almost there's no direct effect. But obviously the changes that are happening in the plants as a result of elevated CO2, it's going to have effect on insects. And obviously temperature has a strong effect on insects. And in many cases, when we look at the elevated CO2, there's lots of different responses like decrease, no change, or compensatory feeding. And that's going to be specific for insect-plant combination. So I've done a bit of work with aphids and viruses and elevated CO2, in this case viruses and bird cherry old aphid, which is the main aphid worldwide that spreads viruses in cereals, and we had non-infected and infected plants under elevated CO2. So you can see here we have decrease in fecundity and elevated CO2 on non-infected plants, but not on infected plants. There was a similar story with Mysis persica, another imported vector of lots of plants, viruses. And what it means is that there is some sort of interactions happening with CO2 and aphids that decrease the population size on elevated CO2 on non-infected plants, but not on infected plants. So what it means is that we might have a greater population of infected aphids flying around in the future. The other thing is the feeding behavior and transmission acquisition efficiency of lots of different viruses, and a similar story that we've seen increased feeding. So even though those aphids had lower population size, the feeding damage will be great. And then we don't have much response with virus infection. And we know now that this is very strongly related to amount of nitrogen in the plants, and aphids respond very strongly to nitrogen in the plants. So the other part is the viruses and how viruses responses. When I first started doing research on elevated CO2 and viruses, it was quite apparent that the symptoms were much more obvious and much earlier. And we've done a few experiments. This one is looking at temperature. So we increase temperature, we grew plants under higher temperature, ambient temperature, and we noticed that there was greater virus titer in plants and elevated CO2. Excuse me, we have still quite a few spare seats here if you want to come in. And the other one was with elevated CO2. It was actually exactly the same story, increased virus titer or virus amount and elevated CO2. So what does it mean is, potential transmission acquisition, greater transmission acquisition efficiency. The other thing was trying to understand, take the opportunities from the free carbon dioxide enrichment facility, which we have this really good facility when we had plots exposed to ambient and elevated CO2. And I collected over quite a few years lots of samples. Over 12,000 samples were collected and were tested for a few different viruses. So looking at natural infestations in natural settings. So we had no control over, when aphids were flying from outside, but what we had infected, we had plots and elevated CO2 and ambient CO2. As you can see here, we have few different viruses, 3 different viruses that were tested in 2013, 14, 15, 16, so a few years ago. And obviously from year to year, there is variation in amount of virus incidence, but you can see here that the black bars represents the elevated CO2 and the other one's ambient CO2. As you can see here, we have over 10% increase in virus incidence under elevated CO2. So what I want to say is that this is extremely critical to understand how plants are going to respond to insect and viruses in the future, and we have to really equip ourselves with some strategies to be able to sort of come ahead of those challenges in the future and be able to protect our food production. So very briefly want to acknowledge a few people there. And also what I want to say is that Grains Research Development Corporation largely funded most of that work, which we're very grateful for. And I think now I'm going to pass to Manuela, who is going to talk about viruses and water stress.
Yes. So hi everyone. Hi everyone. I'm going to speak about the result we got in the lab. So I'm from INRAE in Montpellier, and as said, Piotr just now, abiotic stress can modulate plant virus vector interaction mainly through changes of the host physiology. And so what we can see is that the virus load and virulence of, for example, can be really altered by abiotic stress, like salt when plants are growing under salt condition, or even water stress and temperature. And there are also something else which is very important. It's a vector biology, so which was shown to be also altered, for example, by high temperature with longer longevity, fecundity. And in fact, all these data speculate on an effect on virus transmission by vector. But however, there are really few data showing the direct effect of abiotic stresses on transmission. So when I started this work, the idea was to settle first a pathosystem with a good water deficit protocol. So I use water deficit abiotic stress. So we use Brassica rapa, which is a turnips plant, and with a cauliflower mosaic virus, and we settled transmission experiment. So what we did in the lab, so it was to inoculate mechanically turnip plants, and after all, settle a water deficit protocol, which was really to stop the irrigation until we reach 50% of field capacity in the pots. So we had the two conditions, which was a well water plant infected and the water deficit plant infected. And from this, we did the transmission, like I told you. So we have to know that cauliflower mosaic virus is a virus which is transmitted, so acquired and released into new host plant very quickly during brief puncture when the aphid is feeding on the plant. So we put aphid on infected plants and for 2-minute acquisition, and after we transfer to healthy plants for inoculation period, and after 3 weeks we look at the symptoms. Between we remove, of course, the aphids. So what we see systematically, we did a lot of experiment, and all the time we can see a very high an increase of virus transmission when plants are in water deficit, when those plants are under water deficit. So what can be the explanation for this? So we have two kinds of interaction which can be responsible. It's a plant virus interaction. So we need to look at the virus load, the viral protein accumulation that could explain a higher transmission. Also the interaction between the plant and the vector that can really be a parameter influencing transmission. So we look at the first kind of interaction, so the plant virus interaction. So we did some virus accumulation analysis using qPCR. And we look at the viral transcript, and also at the protein level, we did the quantification of the protein P2, which is really involved in the transmission process of this virus. And apparently, we do not see any difference in these viral traits. So now when we look at the interaction between the plant and the vector, we look first at the feeding behavior of the vector. So there is a very easy way to, not very easy in fact, but there is a way to look at the feeding behavior. which is the EPG technique. So in this technique, in fact, the aphid is connected to an electrical circuit, and it's making waveforms that are really specific of the feeding behavior. So among the patterns that we can analyze and that could explain a better transmission, it would be the number of probes that the aphid is making on the plant, infected plants under well water condition or water stress condition. So in fact, when we did this experiment, we see clearly that there is a significant increase of the number of probes here in the two conditions, which is here the healthy condition but under water deficit, and the infected condition under water deficit. So it really seems that water deficit plants increase the feeding behavior, the function, the number of probes of aphids feeding on them. So that can also explain transmission. And also what we can see clearly is that there is a significant effect of the infection on the fecundity, the body size of the aphids. So the fitness is increased when aphids are growing on infected plants. However, the watering statue. So we went on another pathosystem, which is Arabidopsis thaliana CAMV. In fact, it was in order to see what could be the variable responses of plants under water deficit. So we used 25 accessions of Arabidopsis thaliana, which were collected in various gradient of rainfall in Spain. And we look at the responses to CAMV and the water deficit responses. Well, so here is the protocol, which is basically the same than before, so I'm not going to explain it much more. So we infected the plant, did a water deficit protocol, and we measure several parameters of growth on the plant side, and on the virus side also we look at the transmission and the virus accumulation. So what we can see when we use this, when we did this, is that, for example, the virulence of the, here we have 25 ecotypes. So in gray light, you have the water deficit condition infected, and in dark, the water deficit, no, the well water, in gray light, well watered condition with infection, and in dark, the water deficit condition plus infection. So what we can see is that there is really a difference in virulence. The virulence was assessed by the reduction of the above ground dry mass of the plants. So we have some ecotypes where we are really tolerant to the CAMV infection here, and other ones which are much more susceptible. What we can see also is that when you add the water stress condition, you have an effect even more detrimental for the plants. We look at the virus accumulation also, and in 8 ecotypes we could see that there was an increase of the accumulation when plants were under water deficit, and in 2 ecotypes there was a reduction. So there is an alteration of the accumulation in some ecotypes. In the same way, in transmission, we could see only in 4 ecotypes differences in transmission rate depending of the watering statute. And what was interesting, it's when we did the correlation between 2 viral traits, which is accumulation and transmission. In the well water condition, we can see that there is a positive trend, meaning that the more virus you have in the plant, the more chance you have to have a transmission, which is quite logical. However, when we look in the water deficit treatment, it was reversed. So I will finish with a take-away message. So what we show is that water deficit on Arabidopsis thaliana alter viral trait of the CAMV, and the plant virus response is really host-specific, because depending of the ecotype, as you saw, there was not always the same responses. And the relation also between viral traits are impacted by the water deficit, like we show with accumulation transmission. And when we take the other pathosystems, we can see for Brassicarapa plants that water deficit really increase transmission by vectors, and that the fitness and feeding behavior of the vector are positively altered in a way that could increase, that is increasing virus transmission. So now we have the big question: what could be the epidemiological consequence of this data, since we show that water deficit increase the fitness of the vector, and that the transmission per se is also increased for the CIMV. So how can we use this data to implement modeling, for example? Thank you.
Thank you, thank you, Manuela. Unfortunately, we used up our time, and we really want to stuck to time, so I think we're going to move to the next speaker. So what I'm trying to say is that we have no time for questions. So if you want to ask us questions, please. One question. Okay.
No, no,
actually, we have.
I don't
Okay, we have time for one question.
Yeah, one question.
So for that.
No, still.
So you mainly tested on cauliflower mosaic virus, right? Which is a non-circulative virus.
Yes.
Would you expect differences when you look at persistent or circulative viruses?
Very, very good question. In fact, we did it. So I present the result with the CIMV, but we did also with another non-circulative virus, which is a potivirus, TUMV, and we had the same result, and it was even more concerning the transmission was double. And I did also with a circulative virus, which is a turnip yellow virus. And in that case, it's the opposite. You have a very strong reduction of transmission. And in fact, we looked in the aphid, and in that case, you see that the aphid doesn't feed well on water-stressed plants. So it cannot acquire the virus, because in that case, it's acquired in the phloem cells. So the aphid doesn't feed well on phloem in an infected plant, water stress. It's me. Okay. So if there is no question, now we can invite Larissa ADAMIK. And she's going to speak about Fusarium. You have to, because I don't have access.
Okay. So thank you, Manuela, for introducing me, and thank you for this opportunity to talk about my PhD project here, which is supervised by Ludovic Bonhomme and Thierry Longin, and is all about the characterization of the weed-specific responses to Fusarium head blight and the contrasting irrigation. So just a small glimpse of, or a small reminder for those who are not, for those who are familiar already with Fusarium head blight. So this is a disease that is affecting small grain cereals and is provoked by a fungal complex. And in this fungal complex, we can find one species, that is Fusarium graminearum, which is dominating this complex, at least in Europe and some other regions of the world, and which we are using in the lab to infect our plants and to mimic a Fusarium head blight. Then the problem with FHB is mainly the big yield losses due to grain abortion, but also the mycotoxin production from this fungus. One of those mycotoxins is deoxynivalenol, which you can see right here. And just for you to show you what is the FHB symptoms, here you can see the healthy spike and healthy grains, and in comparison you have diseased spike and diseased grains. So in my team, we are working about 10 years now on the susceptibility determinants on this disease. And FHB is indeed controlled by a range of susceptibility factors, which are overviewed here in the circus plot. And two of the most important susceptibility components, according to our team, are first regulatory hubs that are transcription factors that in turn, okay, there's a bug, no problem. So the regulatory hubs are regulating target genes, but also other transcription factors that in turn can regulate target genes. And the second, component that is really important are chloroplastic processes. That means water and carbon fluxes. And they have a strong link with the pathogen effector regulations. So this is illustrated here in this graph, where we have the abundance dynamics of chloroplastic proteins. And there are big changes occurring between 48 and 72 hours post-infection. And this knowledge is quite powerful because now we can actually assess the remodeling of a response upon environmental changes, and we can really evaluate and characterize how specific this stress combination response can be compared to the single stress response. So the main objective of this project is to identify new components of plant stress responses to multiple stresses that are able to control FHB in fluctuating abiotic environments. So this contains first the evaluation of abiotic stresses on FHB development, and in the second time the assessment of remodeling of the plant responses using dual omics approaches, which in my case are dual RNA-seq and dual metabolomics, which I will focus here in this talk. So as you already understand, my abiotic stress is Drought because it is extremely relevant given that the role of water flows is important in the response to FHB. So what we are doing in the lab is that we're growing wheat until the flowering stage where it is susceptible to the Fusarium graminearum. That's why we are inoculating it at that stage. And the novelty of my project is that we cut the irrigation either 6 days before flowering stage, so we have 6 days of drought, where we have then the installing stress at the flowering stage and an early stress response, and or we cut the irrigation 12 days before, when that permits stabilized stress at the flowering stage and a rather late response. What you can see here is the pot weight evolution during the experiments, and you can see that there is no difference in the intensity, but only in the duration of those stresses. And what you see also is that there is an increase here and here because we are re-watering the plants when they are at flowering stage, so we are sure that there is a successful infection. Then we wait 72 hours before getting our samples for transcriptomics and metabolomics. And during this time, and even 7 additional days, we monitor the symptoms. The symptoms are monitored with a scale from 0 to 4, with 0 no symptoms to 4 with maximal symptoms. And then we calculate the area under the disease progress curve, which you can see here for the different water treatments. And indeed, we have successful infection also in the limiting conditions. And what is really interesting is when you compare the 6 days and 6 days water stress and the well-watered plants, they have a similar disease progress. However, when you compare the 12 days moderate water stress plants, they have a reduced disease progress. And this is the first indicator that we really have a specific response of this stress combination here. So how to characterize this specificity and how do we get this reduced symptoms? So we did a metabolomics approach with the collaborators here in Toulouse. And we did LCMS-MS, and we succeeded to annotate 375 metabolites, of which you can see the distribution of the different families right here. And in this approach, we have the metabolites from the fungus and from the plant. So when we do a parent sample component analysis on this whole data set, you can see that the first dimension is corresponding to the water regime, where we are going from 12 days water stress over 6 days and then to well-watered plants. And the second dimension corresponds to the infection, where we have the infected plants here on the bottom and the mock-inoculated plants on the top. The variables are greatly contributing to the explanation of our variance, and we can conclude from this that the stress combination with each individual stress explains a substantial part of the responses. So, but which are the metabolites that are really interesting in that data set? So we had a first statistical approach with a stress quality model, we're calling it, where we explain the intensity on, based on the stress quality. That means we are comparing no stress condition, drought with no infection versus infection with no drought, and versus the combination of drought and infection. So we're considering our combined stress like a new individual stress. Like if you just had an additional third, like for example, a salt stress, we're just comparing it like that. And this gives us 9 metabolites that are completely specific and unpredictable from the single stresses in the stressor response. So we have, what is interesting is that we have different families that are implicated in that response. And in a second time only, we wanted to find the dependency of the combined stress responses from the individual stress responses. So that we did a more classical approach with a complete model, explaining intensity by the drought and infection. That means we compare no drought versus drought with or without infection versus infection with or without drought and their interaction. And here we get 7 metabolites that seem to be more or less predictable from the drought and the infection responses. So in this talk, I want to focus on the specific metabolites. And what is quite interesting is that we have 3 different metabolites that have this accumulation profiles. One of them is the carbohydrate derivative, one carbolic acid derivative, and a ferulic acid derivative. And however, we don't have an annotated role, in a biological role for this one. The hypothesis that we have in the lab is that it might be linked with the energy supply of the pathogen, as it is a carbohydrate derivative. Indeed, the 2 other ones they are known in the literature, and they were already identified as antifungal molecules. That makes sense because we have them here in the combined stress response, quite highly expressed. And we are supposing that those metabolites might be associated with decreased susceptibility into FHB. However, we of course need to really validate this in plant still. What is also interesting is that there are this ferulic acid derivative. We have the upstream and downstream metabolites of its pathway. So we have the chimeric acid and coniferin in our data that we measured. And in general, we suggest that phenylproteinase are playing a key role in the stress combination. And this makes sense because phenolics in general are involved in various biotic and abiotic stress defense responses. But also, when ferulic acid is used to treat an exogenous treatment, it mitigates salt stress in cabbage, but also in wheat, and it is inducing this, inducing rust scavenging, which might be a key role also, playing a key role in FHB. What is interesting too is that the ferulic acid derivative we are measuring here is documented as an allochemical affecting the seed germination. But why is this interesting? It's because seed germination induces also carbohydrate release and is documented to be closely related to the plant immunity. So to conclude, we showed here that water fluctuation mitigates susceptibility to F. germinarium. We showed also that there is a specific response combining drought stress and FHB, and this specific response might help to reduce FHB symptoms through the regulation of defense molecules, such as those we are showing here, carboxylic acid and ferulic acid derivatives. And what is still left to do is the evaluation on these findings and confirm these findings eventually with RNA-seq data that is already available but that has still to be analyzed. And we want to evaluate the key role of the metabolites I showed you by infusion. So thank you very much for your attention, for your time, and a special thanks also to my supervisors.
Any question? No?
Okay. Yeah, you can still ask questions in the chat. I think I will have a look at it, and you feel free to contact me if you want to.
Okay, thank you, Larissa.
Yeah, thank you very much. We have another talk on plant viruses. Clara LAGO going to present on viruses and drought stress.
Okay, good morning, everyone. Today I will be presenting this work, which title is Drought Exacerbates Virus Effect on Canola Plants. Climate change is one of the major challenges that humanity is facing nowadays. The emission of greenhouse gases have been accelerating until achieving alarming levels nowadays. These have increased heat trapping, which have affected the global temperature, and these have also increased the frequency and the severity of extreme weather events such as hurricanes, floods, and drought. Oh, sorry. Well, these not only impact the global climate, but also have an effect on all living organisms, which include plants, pathogens, and its vectors. Furthermore, the global population has been increasing, so achieving food security becomes a major concern. Currently, plant viruses are one of the major causes of crop losses worldwide, and climate change is only making the situation worse. There are several studies that show that climate change will have a significant effect on plant virus epidemiology. When there is a disturbance on some abiotic stresses, such as the concentration of CO2, the temperature, or the water availability, it may have a negative, positive, or neutral impact on the pathosystems. Among different stresses, drought have emerged as a prominent threat for agriculture worldwide, being a major cause of yield losses. Drought may have a direct impact on plant physiology and metabolism and affecting the defense response. It can also impact the virus titer and the symptom expression, or directly affects vectors behavior. In a work conducted by Basket et al., they saw that drought and increased temperature increases potato yellow vein virus concentration, boosting symptom expression. On another work conducted by Maraconda, they saw that drought increased the mortality on Arabidopsis thaliana infected with turnip mosaic virus. On the contrary, other studies show that drought may reduce the transmission of turnip yellow virus by Mycos persicae. And on another work, they saw that barley yellow drought virus infected plants recover from severe stress more readily than non-infected plants. All this literature review shows that the interactions between plant viruses and drought stress are complex. So focusing on our work, canola plants is a major oilseed crop cultivated worldwide, and canola crops are highly susceptible to drought stress at different growth stage. Turnip yellow virus is an aphid-borne circulative virus that causes great yield losses on canola crops. So the objective of our work was to investigate the interactions between turnip yellow virus infections and drought stress on canola plants. In our experimental design, we subjected the canola plants to 3 different water treatments. The well water treatment, in which we water the plants until achieving 100% of the field capacity. The water stress treatment, in which we water the plants until achieving 60% of the field capacity. And the terminal drought. The plants were well watered, but we stopped watering them at some point. Then we also have infected plants using Mycetes persicet as the vector with turnip yellow virus and non-infected plants. We directly shown the plants on the pots, and then we inoculate them with the aphids. After 14 days after the inoculation, we started the irrigation treatments, and during the whole experiments, we continuously measure several different variables that shows the growth of canola plants. We also perform one destructive sampling to take some destructive measurements, and at the end of the cycle, we collect all the plants. Between the first harvest and the second harvest, we stop watering the plants from the terminal drought treatment. The variables that we measure were the dried weight, the leaf area, the water used by the plants, the plant height, the chlorophyll content, the number of leaves, the number of inflorescence, the number of flowers, and the turnip yellow virus symptoms. We also calculate the additive ratio to explore the interactions between the drought stress and the virus stress. When this ratio was higher than 1, that means that the combined stresses are higher than the sum of the stresses itself. When the additive ratio was 1, that means that the sum of both stresses is the same as both stresses combined. And when the additive ratio was less than 1, that means that the effects are antagonists, that the sum of both stresses are less higher than the both stresses combined. We performed 2 different trials, a competition assay in which we have 4 plants on the same pots, 2 infected and 2 non-infected, subjected to the different water treatments. And another assay under non-competitions, the plants were in a single pot, and we have infected plants and non-infected plants subjected to the different water treatments. Then I'm going to present the results of the competition trial. We observe that when we compare infected plants with non-infected plants, the dry biomass was significantly lower on infected plants on the 3 water treatments. We also observed that for non-infected plants, the dry biomass was significantly lower on the under a water stress regime compared to well water conditions. And we observed the same for the plants that were infected. And by calculating the ratio, we observed that the interaction between both stresses is additive. Similar results were obtained for the leaf area. The plants, when were infected, the leaf area was significantly lower compared to non-infected plants for the 3 water regimes. We observed that for non-infected plants, the leaf area was significantly lower under water stress conditions and terminal drought conditions compared to well water conditions, and the same for infected plants. And also the effects between virus infection and water stress is additive. For the number of inflorescence, we again observed that for infected plants, the number of inflorescence was significantly lower than for non-infected plants on all the water treatments, and that the interactions between both stresses are additive. But for the flowers, the number of flowers, we just found that when plants were infected with turnip yellow virus, the number of flowers is significantly lower compared to non-infected plants when they were under water stress. With the other variables that we measure, we observed that the water use was higher on well-watered plants. The plant height, we didn't find significant differences, neither for the number of leaves. We didn't observe clear virus symptoms, and for the chlorophyll content, we observed that was higher on water-stressed plants and terminal drought plants compared to well-watered ones. Overall, with this trial, we observed that the growth of canola plants was substantially impaired when plants were subjected to combined drought stress and turnip yellow virus infection. For the results of the non-competition assay, the results were similar. For the dry biomass, we observed that when plants were infected with turnip yellow virus, the dry biomass was significantly lower than for non-infected plants under water stress conditions and well water conditions, with no differences with the terminal drought. We also observed that for non-infected plants, the dry biomass was significantly lower when they were under water stress and terminal drought conditions compared with the well water conditions, and the same for infected plants. And also, the sum of both effects were additive. With the leaf area, we also observed that under water stress conditions and terminal drought conditions for non-infected plants, the leaf area was significantly lower than under well water conditions. The same with infected plants, but we didn't find differences between infected and non-infected plants in any of the treatments. With inflorescence, we also observed that infected plants have significantly lower number of inflorescence than non-infected plants under the 3 water regimes. And the same situation was observed for the number of flowers. Also, for the number of inflorescence and the number of flowers, the effect of virus infections and water stress are additive. With the other variables, we observed that the water use, again, was higher on well-watered plants, that the plant height was higher also on well-watered plants, with no differences between infected and non-infected plants. We didn't observe significant differences on the number of leaves, and we didn't observe either clear virus symptoms. And for the chlorophyll content, again, we observed that it was higher on water-stressed plants and terminal drought plants compared to well-watered plants. Again, the results are similar to the previous trial. Overall, the growth of canola plants was impaired when subjected to combined water stress and turnip yellow virus infections, although the effects or the impact is less noticeable than on the competition assay. So, to sum up, both plant growth and flowering was reduced when plants were subjected to both stresses, drought stress and turnip yellow virus infection, compared to non-infected plants under well water conditions. Turnip yellow virus infections and drought stressors combined reduce the growth and the flowering in the same proportion as the sum of the single effects, so there is an additive interactions between these two stressors. And the impact of turnip yellow virus infections and drought stress on canola plants was more severe when plants are competing by the resources compared to non-competition conditions. All these results combined, what shows is that under future climate change scenario, drought combined with virus infection may exacerbate yield losses on canola crops. Finally, I want to say thank you to my colleagues from Agriculture Victoria Macquarie University, especially Piotr, who is the chair of this session, and all my colleagues from the Spanish Research Council. If you have any questions, I will be happy to answer.
Thank you, Clara. We have time for a few questions.
Hi, I was wondering about the genetic context that you use, because some varieties can manage very well drought and others less. And so maybe the results could be completely different depending on the variety. Have you tried to look at that?
No, we didn't explore the genetics, but of course, and as the Piotr and Manuela have spoken on the previous session, it depends on the pathosystem and on many different variables, the impact of the combined stress on plant diseases.
Thank you very much for your interesting talk. I assume that you have used spring canola, I think, yeah. Because in Europe we have mostly winter canola, and when you are doing such a kind of experiments, you have to do also the vernalization to have the flower at the end. I'm wondering if you have in a spring canola also resistant cultivar to turnip yellow virus, and if you check if the drought stress or higher temperature had the effect on the overcoming of the resistant, for example.
We didn't explore that. These experiments were performed in Australia, but definitely it would be very interesting to check on that. Thank you.
Any more questions? I have a very quick question. So you mentioned that there's few studies showing that in some cases a virus can protect plants from drought stress or other conditions. Obviously, this virus is highly pathogenic, so actually cause disease in Australia and whatnot. What's your opinion on the fact that other studies showing otherwise? Would you see this virus actually in any cases combined with temperature and drought have any capacity to help the plant grow under those conditions?
Of course, the stressors like higher temperatures, drought, have a huge impact on yield losses as well as the pathogens. Reviewing the literature, most of the studies shows that in a future climate change scenario, the situation is going to be even worse. But there are some studies that shows that maybe these complex interactions may not help, but maybe reduce the impact of virus infection. So I have not a specific answer. I think it depends on the pathosystem and on the environmental conditions. But generally, the situation is worse under both combined stress.
Do we have another last question? Yeah, there's one.
Nice and interesting presentation first. Did you check the phytohormone status in those situations?
Sorry?
Phytohormones status, particularly ABA, under your progressive stress.
No, we didn't check, but it's also something that worth to analyze, because under drought and, under virus infection, sometimes there are some phytohormones like acetic acid and things like that that may have also impact on the resistance to drought. So it's worth to analyze that too, but we didn't do that.
Well, thank you very much, Clara. Please help me thank Clara again for her presentation.
So now we go to the next speaker, which is Antoine DAVIERE.
Okay, good morning everyone. I'm very happy to be here today to be able to present you part of the work I've done during my PhD. And I will thank deeply the organizer to have given me this opportunity. So I'm going to talk firstly about the first characterization of pectin lyase in Botrytis cinerea that is involved in pathogenicity. And secondly, I will explain you how this particular pathogenicity factor contributes to the impact of host nitrogen nutrition on disease severity. So Botrytis cinerea is an ascomycete of the Sclerotiniaceae family. It's a necrotrophic fungus that is responsible for gray mold disease on more than 1,000 host plant species. So it is, this fungus is well known for the large diversity of secreted pathogenicity factors. Here you can see cell death-inducing proteins, protease, the production of reactive oxygen species, and a small interfering RNA, but also toxins and metabolites, and a large diversity of cell wall degrading enzymes. So today I will focus on these cell wall degrading enzymes, and more particularly pectin degrading enzymes. So Botrytis has different types of enzymes that can degrade pectins. There is first pectin lyase that are enzymes that are able to degrade the homogalacturonane polymer in its highly methylated and acetylated form. Whereas polygalacturonase are able to degrade the polymer after its desesterification by pectin methylesterase and acetylesterase. In Botrytis, there is a very abundant production of polygalacturonase, and they are well described as very important pathogenicity factors, whereas nothing is known about pectin lyase. So this degradation will produce what we call oligogalacturonides that are, or OGs, that can be differentiated by their origin because there is a specific chemical signature of pectin lyase degradation, that is, it's a double bond terminal galacturonic acid. And the OGs are well known for their activity as danger-associated molecular patterns that are recognized by the plant and can induce PTI. In a previous work with my team, Aline Voxer set up a technique to detect oligogalacturonides during infection by high precision size exclusion chromatography coupled with mass spectrometry. And this technique enables the separation of OGs by the degree of polymerization. As you can see here, large OGs have shorter retention times than short OGs. And it also enables the separation by the number of methylation. Here you can see that Gale A3 have a shorter retention time than its methylated form. This work also showed that during infection of Arabidopsis by Botrytis, 80% of the OGs produced are derived from a pectin lyase activity, indicating a potential role in pathogenicity. Furthermore, they showed that an OG mixture enriched in long OGs derived from pectin lyase of DP above 4 is able to induce the expression of a repressor of the GA pathway. So the hypothesis was that those particular OGs are able to repress defenses in contrary to the other OGs that are known for inducing defense. So during my PhD, I've worked on the several pectin lyase gene of Botrytis. So there is 5 homologs and 4 that are expressed during the infection. Here you can see their expression in 1 day post-inoculation and 2 day post-inoculation. And you can see that BCPNL1 is strongly expressed during the early infection, whereas the other homologs respond later. So that's why we have focused our attention on this gene. And so for that, I've constructed independent and homokaryotic mutants with the CRISPR-Cas9 technique that was adapted recently on Botrytis by the group of Matthias Hahn in Germany. And so I've obtained 3 mutants and tested them on Arabidopsis. And as you can see on the picture and on the graph, those mutants have a reduced pathogenicity compared to the B0510 reference strain, indicating that this pectin lyase is involved in pathogenicity on Arabidopsis. I then tried to test this mutant on tomato, and we found the same results, indicating that this gene is not only restricted to the importance of this gene on pathogenicity is not restricted to one particular host plant. Then I studied if this gene can play a role in plant defense regulation, and particularly in the GIA pathway. And so here you can see the expression of a GIA repressor and a GIA marker during the infection with the mutant and the wild type strain. And we see a lower expression of a GIA repressor that correlates with a higher expression of a GIA marker with the mutant, indicating that this gene is involved in repression of the GIA pathway during the infection. There is a problem. Here is a heat map of all the OGs. So, sorry, it was okay before, but I don't understand. So the message was that we studied the OG production during infection with the mutant, and we found that it was very striking that there is almost no OG production with the mutants. So you can't see, but you have to believe me. And so this result indicates that PNL1 is a key enzyme of the pectin degradation machinery of Botrytis. Then the second part of my project was to study environmental conditions that impact plant diseases, and more particularly the study of plant nitrogen nutrition. It's well known that plant nitrogen nutrition impacts interaction between the plant and a pathogen, and this interaction is highly dependent of the pathosystem. Sometimes high nitrate nutrition induce a stronger disease, and in other cases it's the contrary opposite. Higher nutrition prevents the apparition of the disease. But in our specific pathosystem, a previous work of my team showed that high nitrate nutrition increases the disease severity. And the mechanism was studied, and we found that the expression of defense gene is well affected by plant nitrogen nutrition, and in particular glyphosate and salicylic acid related genes. And it was surprising also to see that a lot of pathogenicity genes are affected by plant nitrogen nutrition. Also, we studied the apoplastic content that is impacted by nutrition. And you can see more detail about that on a poster presented by Mathilde Fagard. So on my topic of on pectin degrading enzyme, I found that pectin lyase, and especially the pectin lyase 1, is more expressed in low nitrogen nutrition. And it was the same for pectin metylesterase and polygalacturonase. And so we wondered if these differences in pectinase expression could affect the OG production during infection. And indeed, here you can see the proportion of long OGs derived from pectin lyase over time in high and low nitrate. And you see that there is a higher production of those GR repressing OGs during the early phase of infection in high nitrate nutrition. I've also studied the effect of PNL1 mutation on N regulation of plant defense gene during infection. So here you have a heat map showing the log2 fold change between high nitrates and low nitrates. During infection with the wild type, you can see that nitrogen really impacts the expression of the gene tested, whereas with the mutants, we almost completely lost regulation by nitrate of this gene. So indicating that PNL1 is also involved in nitrogen regulation of plant defense. So here is my conclusions. We found that PNL1 is a key player of the early pectin degradation by Botrytis, and that it is also involved in the effect of plant nitrogen nutrition on the interaction. So here is a small model of the results. We found that at low nitrogen, we have an effect on expression of the pectinase that induce a high turnover of the large PNL1 OGs to obtain a strong induction of defense and the GR pathway. Whereas in high nitrogen nutrition, we have a slower turnover of this long OG products that are able to repress the GR pathway that can explain partly the differences in disease severity induced by nitrates. So now it will be interesting to find the signals that are responsible for N-regulation of pectin-degrading enzymes. So I would like to thank my team and also all the people that I've worked with on this project. And I thank you for your attention, and I can answer. I can try to answer your question.
Okay, any questions?
Thank you for your presentation. What do we know on the effect of this nutritional status on Botrytis fitness in general? Do we have any idea? Because it is written that Botrytis feeds on pectins. It has been written often. But what do we know in terms of the fitness when we grow Botrytis or we inoculate on those high level, low level nitrogen?
You mean in vitro?
Yes, it could be in vitro.
The level of nitrogen really impacts the production of pathogenicity factor in a large different range of fungal species. But we have studied in the plants that are grown with high or low nitrogen nutrition, that the content of nitrates is not so different and cannot explain the differences that we saw in terms of expression of pathogenicity factor. So it's not the whole picture. It cannot be explained just by nitrate nutrition of the pathogen.
More question? No. Okay, so I let you.
Okay, so now we're going to be moving to temperature adaptation and bacteria. And Carla Cardenas Gomez is going to tell us more about it.
This was too high for me. Good morning, everyone. I'm Carla. I'm a first-year PhD student. And today I'm going to talk about the question I'm aiming to answer during my PhD, which is, can temperature adaptations drive Ralstonia solaraceum range expansion? And I'll start with this image that is captivating to me. That is Ralstonia solanacerum. And this image is interesting to me because it comes from this paper, which is very interesting. It's a top 10 of the most dangerous bacteria plant pathogens in the world. And in the top 2, Ralstonia over here is there. So bacteria is the second most dangerous bacteria plant pathogen in the world. And the reason why is because Ralstonia has a wide host range. Around 300 species of plants get affected by this: tomato, bananas, tobacco, peppers. So it's very important in agriculture, and it causes wilts and rots. So in the image here with the potato, you see a potato with a ring of bacteria oozing from it. And then in the other image here on the right, you have a bacteria with a plant with the symptoms of bacteria wilting. So Ralstonia is a soil and waterborne bacteria plant pathogen. And once in the soil, the bacteria goes through the roots of the plant, and once in the roots, goes through the xylem to the more aerial parts of the plant. And once that produces a mucoid kind of substance that is called an exopolysaccharide, blocking the xylem system, and then the plant loses its turgency and dies. Another important characteristic of Ralstonia is that it's widely spread in the world. So it's in all the highlighted areas in this map. So also interestingly, Ralstonia is very diverse genetically. So it affects hundreds of species of plants, and it also survives in different environments. So there are hundreds of strains of Ralstonia that have been grouped in the Ralstonia solanacearum species complex. This complex has been divided into 4 phylotypes, and they are warm-adapted and cold-adapted strains. In this table, you can see the 4 phylotypes, and the phylotypes have been organized based mainly in the origin, in the geographical origin. And today, I'm going to talk about phylotype 2b. There are many factors that might contribute to the spread of Ralstonia in so many places in the world. And one of those is the secondary hosts. So Ralstonia can also have secondary hosts. And one of those is Solanum dulcamera here, this plant here. This plant is a weed that is commonly found in Europe in the riverbanks. So as I mentioned before, Ralstonia is a waterborne and soil-borne. So this plant is around the river. So Ralstonia goes to the roots of the plant, survives there, doesn't cause any symptoms. When the temperature increases in the water, there's a spillover of the bacteria, and then that water, of course, cannot be used for crop irrigation. Now, the mechanistics behind how Ralstonia doesn't cause symptoms in secondary hosts is not completely clear. Another very important factor is temperature. In laboratory conditions, 28 degrees is the optimal temperature for Ralstonia, but also temperature has a huge impact in soil persistence. Ralstonia can survive up to 7 years in soil, and it is important for not only soil persistence, but also causing symptoms in plants. So there are some studies reporting that resistant varieties of tomato, for example, when they're subjected to high temperatures when inoculated with Ralstonia, they start causing wilting symptoms. And finally, we come into the equation, so human agricultural activity. So there have been some incursions of Ralstonia phylotypes in different areas of the world because of the movement of infected plant debris, of tools, or agricultural tools. So we have a problem here that's kind of like a puzzle. So we have global warming, increasing temperatures, plus the human impact in agricultural activity. And then the ability of Ralstonia to adapt to different environments that might create new and more aggressive strains with the ability not only to spread in other areas of the globe, but also maybe to cause infections in secondary hosts. So again, we have many pieces of the puzzle that are not completely clear. So the aim of this project is to assess if cold-adapted strains can evolve and adapt to survive at warm temperatures in light of global warming. So the first question to answer here is, do cold-adapted strains can survive at warm temperatures? And if so, these lead to generalism or specialism. Generalism meaning the bacteria can survive at a wide range of temperatures, or specialism just surviving a specific range of temperatures. And finally, and most importantly, if this will have an impact in Ralstonia's virulence, and if this might have an impact in secondary hosts, this will have an impact in agriculture later. To tackle these questions, I have been using in vitro experiments, but of course, because this is my first year, I haven't done yet the in-plant experiments, which is something I'm planning to do later, with both primary and secondary hosts. Oops. Oh, there was an image here. Well, you have to believe me, but this image here was a Petri dish with wire-199. Wire-199 is a bacteria strain isolated from the UK near Oxford, and it's a phylotype 2b that has the ability to survive at low temperatures. The treatments I have been choosing to study these bacteria have been 15, 20, 28, and 34. So it was performed as serial passage experiment. So we had wire-199, this UK strain, at liquid media. And then the fresh 24-hour culture was plated in 96 well plates by 4 replicates. So 10 microliters of the bacteria were mixed with sterile media. And then each replicate was submitted to a different treatment temperature, 15, 20, 28, and 34 degrees. They were incubated for 3 days. After the third day, the optical density was measured. And then a serial passage was conducted. 10 microliters from the previous inoculum here was moved to fresh media. And this process was repeated 12 times. So this experiment took 39 days, 12 passages. Oh, sorry. My plots are not here. Okay. Well, here I'm happy to share the presentation later. So I had a plot, I guess. During the treatment, and then after 20 degrees, so this will be 15, 20, 28, and 34 were something like this. So at 15 we didn't see variations. Then at 20 we saw an increase around day 15, which it was similar to 28 and 34, but then we saw a drop in the optical density or the bacterial density. So this was interesting because this bacteria, that it was usually adapted to cold temperatures, was showing an increase in bacterial density at high temperatures. That led to this question that you actually can see here. That is, if this bacteria was a generalist or a specialist. So to answer that question, we perform a fitness assay. So from the last passage, so from the last point time taken from this experiment, we did CFU per mils. So we took colonies from each evolved community at each temperature, and those colonies were mixed with liquid media, and then incubated at 28 degrees for 2 days. 4 replicates were done to submit it again at the initial temperatures, 15, 20, 28, and 34. Then the optical density was measured, and here we have the plot. Yay! So each graph here, so we have each treatment, 15 degrees, 20 degrees, 28, and 34. Each line corresponds to the evolved populations, the populations that were evolved at 15, 20, 28, and so on. The most interesting result was the treatment at 34, because as you can see in the blue line, is the communities that were evolved at 28 degrees. And then they're growing better at 34 degrees. But then they have a trade-off result because they're not growing as well in all the temperatures, in lower temperatures, sorry. Meaning that there was a trade-off, meaning that there was an adaptation, meaning that maybe this strain can adapt to evolve to survive at higher temperatures. And this has an important impact because that means that cold-adapted strains can adapt to survive at warmer temperatures in like the global warming, and that can maybe establish. And if there is an incursion from Y0199 to another parts of the globe, maybe there can be an outbreak of this, which is very interesting and important. Again, I have only done in vitro experiments, and I'm hoping to do in-plant experiments to corroborate these results. Future work, we would like to do the other way around to test if warm-adapted strains can adapt to survive at cold temperatures. Of course, do all the genetic analysis of these results to test if the evolved bacteria can, where the changes, where the sequencing changes, and compare those results. And finally, and the most important part, will be to test the virulence, to test if these evolved strains have an impact in both primary and secondary hosts. I want to thank my supervisors, Andrea Harper and Wille Freeman, and all the team, and the University of York, the Department of Biology, and thank you for your patience and these technical issues. Thank you.
thank so thank you, Carla, for this marvelous and very interesting and enthusiastic presentation. Sorry about these technical issues. We had a chat with people there trying to fix it up. So hopefully, next sessions we don't have the same problem, and we have definitely time for questions. So you have a question?
Thank you for your presentation, and thank you the way you adapted to the. I like your last slide when you show the next steps, and I want to ask one question around the virulence. It is planned already, but do you know if from the literature there is ideas of how bacteria, including the species or other species, can modify, change the pattern of virulent factor according to different external situation, including the one you're working on?
Yes, in literature, in other bacteria, when the temperature increases, there's modifications in the virulence factors having an impact on the host pathogen relationship. That's something I will definitely like to explore after my experiments. I know that, for instance, there have been previous studies with Ralstonia, other strains at 20, 24 degrees, and there are definitely changes in the virulence factors. But again, I'm happy to do it later. Thank you.
Do we have any other questions?
Yeah.
Yep.
Okay, so you said that your strain can resist to low temperature, but did you try did you look at if your strain can survive at 4 degrees? Because, I mean, some other phytopathogenic bacteria like Pseudomonas or even Pectobacterium strains, they can really well survive, and even the Pectobacteria strain can grow at this temperature.
No, I haven't done that, and I would love to do that. And that's the next step, to test those adaptations at cold temperatures. My first pilot study was at 10 degrees, and they were still growing. They were not dead. They were not, I cannot say they were virulent because were in vitro studies, but they were definitely still alive. So I presume that at lower temperatures, some strains will still survive. Yes, you're right.
Okay. More questions? Yeah.
The strain you work with is called adapted, but it seems to me that it grows better even at high temperatures than the others.
Yeah, which is very interesting. So yeah. I don't know. I'm hoping to explain that over my later experiments. We know that it's called adapted because it was in the UK, which is cold, and it was isolated from potatoes. And previous colleagues who did work with that, they mentioned that it is called adapted. I haven't done specifically the gene studies to explain where are the changes to corroborate which part of the gene can explain that cold adaptation yet. But yeah, it's interesting that you have to believe me on this graph that it was growing better at 20, 28, and 34.
Do we have more questions? I have a quick question. It's very interesting that this bacteria is like such a complex and so diverse, and it's distributed across basically the whole globe. And given that obviously we have so many different strains, so many different adaptations and whatnot, and obviously given changing climates, different conditions, one strain might be more prevalent than the other one. So how do we tackle that as an issue moving forward? Like, what kind of information do you think you need to be able to protect plants or predict which strains are going to be more prevalent? I guess we have pretty good understanding of the strain distribution now across the globe in different conditions. Would you be able to use that and plug into a model to be able to sort of, for example, country like Australia, which is hot and dry, compared to, let's say, northern country in Europe, which could be cooler and warmer, to sort of help them, come up with some sort of solutions and have real sort of understanding of the biology and what the cultivars that need to be protected need to have in the future.
That all those ideas sound very good, that it will be a way to model how to react in the future. As previous talks, that it's better to start planning to deal the problems for the future than the problems from the past. I think this is the approach. Also in my team, people work with phages, so this could help to design phages that are specific for different strains, or a cocktail of phages that can tackle different strains depending on the location where they are, and maybe have more specific biocontrols ways to tackle this pathogen and the different strains.
More questions for Carla? Yeah, there's one over there.
I wonder if generation after generation, because it's in vitro culture, you do with Ralstonia, Ralstonia lose its virulence, and how do you think you can control this effect?
Yes, it will lose virulence, probably because it will have a cost. Those adaptations have a cost, so that will be the trade-off. I'm more interested in the points where they become more virulent, or they become more adaptable. Check those genes, do the fitness assays in the plants as well, and check the virulence in that point when they reach their peak. I'm not sure if there is a way to avoid those trade-offs over time, because over time the bacteria loses its ability to cause diseases, and we also have seen that sometimes after passages we see morphological changes, meaning that they're not producing that mucoid substance that blocks the xylem. So it is, at this stage, I don't know if I can predict how to control that, so I'm hoping just to study at the point where they're the most virulent, if that makes sense.
Do we have one last question? If not, please help me thank Carla for this great presentation. And sorry about the technical difficulties.
Okay, so now we go to our last speaker, Christina Steidler, who is going to speak about Drought and the Fusarium head blight.
Good morning, everyone. So I will talk to you today, or I will present our work that we've done in barley, in spring barley. And don't worry, this is a pretty bioinformatic heavy talk, but I will try my best to guide you through all our experiments. Well, we also have climate change happening in Bavaria. So here you should see this famous striped picture starting from blue to more and more increasing red colors. So we are also facing increasing temperatures and climatic change in Bavaria. And more or less at the same time, we also observe in different samples from Bavaria, in different barley samples from Bavaria, an increase in the amount and in the amount of Fusarium DNA contaminations. So here we quantified the amount of fungal DNA in different seed samples from different years. And as we've already heard earlier before, Fusarium is the cause of Fusarium head blight disease in cereals like wheat, but also in barley that you do not see on that slide. So in the frame of this project, we wanted to identify how drought stress affects the Fusarium head blight disease in different barley varieties. And in the frame of this project, we were especially interested in different barley cultivars because we want to be prepared for climate change, and we want to offer for breeders varieties or traits that they should look for in the future. So we have chosen 3 different spring barley varieties called Barke, Morex, and Palmella Blue, and we already knew before that they have a different range of susceptibility to Fusarium head blight. We infected them with a Fusarium culmorum strain, and our experimental setup was done in a way that we stopped irrigation 7 days before flowering, and then we spray infected our plants. And in order to facilitate an equal infection in our well-irrigated and also in our drought-stressed plants, we bagged the spikes in plastic bags so that we really can make sure that Fusarium is growing well or equally in all of our samples. And then after 2 and 4 days, we sampled them. For our transcriptomic analysis, we performed 3' RNA sequencing, which is way cheaper than conventional sequencing, but is sufficient for our questions. And then to analyze this huge data set, we performed weighted gene correlation network analysis, and in the end also calculated a gene regulatory network. And most of the results that I'm presenting today are already available on bioRxiv, and hopefully very soon are also published. So when you do a big experiment like that, you want to make sure that everything works correctly, right? So to monitor the infection success, we measured the fungal DNA in each sample using qPCR, and we could establish that, again, Palmella Blue was our most susceptible variety, shown here, having the highest load of fungal DNA. There should be 2 more rows, one green showing the phytohormone abscisic acid that we have measured in our samples as a proxy for drought stress. And what you should see here is that Barke has the highest content of, or the highest amount of abscisic acid, meaning that it probably suffers the most from the drought stress or has the highest response to the drought stress, whereas Palmella Blue is almost, or is not really showing high amounts of ABA. Of course, we have also calculated differentially expressed genes and also done geoterm analysis. Overall, what we can observe throughout the whole experiment is that all genotypes are more resistant after drought stress. So this was kind of a surprise. So you would probably expect that if a stressed plant is infected, they will probably more susceptible. But in this case, all genotypes showed a reduced amount of fungal DNA after they have suffered drought stress before. So this is all nice and good, but we have over 12,000 differentially expressed genes. Where do you start? What do you take? What do you learn from that? And in order to tackle that question, we performed the weighted gene correlation network analysis. So if we have used our RNA-seq data, you calculate expression profiles, you do a correlation, a pairwise correlation analysis, and this then leads to a weighted network. And based on this weighted network, your genes are put into different modules which show a similar co-expression pattern over all your samples. And the really nice thing about this package is that you can not only do this with your transcriptomic data, but you can also correlate, for example, the amount of fungal DNA with your transcriptomic data, or in our case, we have also measured several phytohormones that can also be correlated with those modules, and then you get a feeling which of your modules and which of your gene sets are probably the reason or correlated with fungal DNA, and therefore might be interesting to look at in terms of infection or drought stress. And we get, when we have done this analysis, we are getting 18 different modules. And I'm also showing you here now 2 examples, the fungal DNA amount and abscisic acid content. And in the case of DNA, we have only one single module called the blue module that is highly significant correlated with the amount of fungal DNA. And this suggests that the genes that are expressed in this module are related to the infection. In addition to that, we have several modules that are significantly associated with ABA, and it's suggesting that those modules are related to the drought response in our plants. And I want to point out here that we have 3 modules, the green ones, that have a positive correlation, and 2 modules that have a negative correlation to ABA, which means that especially in the turquoise module, we find a lot of genes that are actually downregulated, not upregulated. One other nice thing that you can do using this WGCNA package is that you can also do something called binary traits. So you're telling what are my infected samples and what are my uninfected samples, or what are my drought stress samples and whatnot. And then you can also see that some modules are indeed correlating with infection. And okay, of course, this is not surprising, it's also the blue module that's correlated with infection. We also get an overlap with the drought response. But interestingly, there is no module significantly correlated with under drought and infection with any of our modules. So this suggests that there is a modular composition of the double stress, and we see in the whole experiment that most of our responses are dominated by the drought response. So, but still, what's in those modules? So of course, you want to know what are the genes that are in those modules and what are they doing. So we are looking now at genes that have the highest co-expression values, and the co-expression value is encoded in the edge weight of the networks. So I've chosen arbitrarily the 25 highest edge weights, and we end up with a network like this. So I'm showing you here the blue module, so you remember this was the one that was significantly associated with fungal DNA. And to our big surprise, we found in the middle of this network UGT glycosyl transferase, and it is already described in literature that this transferase is important for the detoxification of DON, so of mycotoxin of Fusarium, Fusarium. So this was really nice, and also all the other genes that are surrounding this glycosyl transferase, a lot of them are well described in literature to have important roles in, during infection. So this seems to be pretty nice. And we've also, I'm also showing you here for the green module, so this was significantly associated with ABA. And the proteins that we find here, or the genes that we are finding here, they have all roles in dehydration and in early ripening. And of course, early ripening is kind of a response to the drought stress before. So this is, makes sense, this is all nice. But still, we do not know what are the main drivers of the network and how are those networks regulated. So in order to answer that question, we calculate a gene regulatory network, again, based on our transcriptomic data. And you will get something like this. Well, this is, again, not really helpful because you do not know what do you do there, what's important here, where are the central hubs. And in order to answer that question, we calculated something which is called betweenness centrality, and this is based on shortest path. So that means between any 2 points or 2 nodes in this network, you will find a shortest connection throughout the network. And then if you take all those shortest connections and see through which nodes do those shortest connections go through, you will find nodes that contain a lot of those shortest connections. So that means if you then, for example, color code here the betweenness centrality in this network, you will see that the 2 dark blue nodes are, according to betweenness centrality, the most central ones. And the further you go outside, the lower the score are, and the less central are the genes. So this is pretty nice. So I calculated this for all of my genes, and I plotted them here for my infection data. So on the y-axis, you have the betweenness centrality score, and the higher the value is, the more central is a gene. And I was especially interested which genes are now upregulated during infection. So I plotted on the x-axis the log2 fold change for the 3 different varieties, Barkemorix and Palmella Blue. And especially in the case of Palmella Blue, we find 2 outstanding transcription factors, transcription factor 1 and transcription factor 2, that have both a high log2 fold change as well as a high betweenness centrality. So they really seem to be important in this infection-related network. Those 2 transcription factors, they belong to 2 very well-described transcription factor families, and especially transcription factor 1 is targeting really obvious genes. So among the highest is a glycosyltransferase, a tamartin-like protein, a glutathione S-transferase, as well as a receptor-like kinase. So, of course, this now needs to be validated, so we would like to perform promoter activation assays to validate that there are indeed those activation and interactions are happening. But is it all good? Are we well prepared for climate change now? All our genotypes that we have tested are more resistant? Probably not, so this is not the end of the story. We have performed a second stress combination experiment where we have now applied drought stress and infection at the same time. So you remember in our first experiment that I've just presented, we got enhanced resistance when we applied the drought stress before. But if we apply now drought stress and infection at the same time, all of our tested genotypes show an enhanced susceptibility. So there needs to be work done. This experiment is currently in the freezer, and most of the RNA is extracted and soon to be sequenced and analyzed. And this already brings me to the end of my talk. I would like to thank my supervisor, Ralf Hückelhofen, for giving me the opportunity and the trust to work on this project. He's a very great and highly supportive supervisor. And I would especially like to thank Felix Hoheneder, a PhD student in our lab and soon to be postdoc, who did all the the greenhouse experiments as well as all the lab and wet lab work. And I would like to thank you for your attention.
So thank you for this very nice talk. Any question in the audience?
Thank you for the talk. I have one question on the samples. What do you sample? Do you sample what? Do you perform the experiment on leaves or on the head itself?
Sorry, I should have explained that better. We have performed everything on the head only. So the inoculation was done on the head, on the spikes, and we only sampled the spikes.
I have a question here.
Yes.
So it's very interesting and a very nice talk. So I'm not sure if I understood right. So you mentioned that you're showing the evidence that the Fusarium head blight is lower on plants exposed to drought. Does it mean that because there's lower biomass, or just because there's less Fusarium? And so my question is, if we have, can you speculate why it is? And for example, if we have consecutive drought, would that mean is that we have less inoculum in a field just because there's less production of the spore on the volume of plant, or just because there's less plant, like a lower biomass?
Mm-hmm. So there were a lot of questions now. First, sorry, what did we start with?
So why do you think it's happening that you have increased resistance to Fusarium head blight in barley under drought?
So we think that this kind of early, so we only have this when the drought stress is happening before, right? So this kind of early drought stress is like a stress response, and you can think about this as a kind of priming event. So the plant is getting into kind of an alert state, and this maybe facilitate the observed resistance that we see. But we do not know that for sure yet.
Mm-hmm. So yeah, so how would you tackle that? Because that's sort of interesting findings. I don't know, because I think for other pathogens, I don't really know much about fungal pathogens, to be honest, but I guess, is it something to do with different critical, I know, temperatures or humidity that I need to, to thrive, or just matter of that, plants maybe just put more energy into surviving rather than growing because they don't have the resources?
So we haven't observed any growth-related phenotypes, and especially as we are working with the spike of the barley plants, and this is the most valuable organ to the plant. So this tissue is highly protected. So we observe drought stress responses in the lower leaves, for example. We see that they are getting earlier senescent, but we do not observe any differences in the spikes, as this is such a highly protected tissue. I would assume that this drought stress event before changes already gene expression, and maybe also have some influence on epigenetics, so that the plant can better and easier more respond to the Fusarium infection, and that this causes the resistance.
And I have a remark about it, because I think it's very interesting, the timing of when you apply the draw and after the infection, or vice versa. And you could maybe test this priming theory in your case. So by doing the draw before and after re-watering and after do the infection, so to see if you have the memory of the plants that is really doing the priming. It's something.
Yes, this is definitely something that would be really nice to be doing. Also, what's happening if you have a drought stress event, probably in very young stages of the plant. So is the plant able to keep up that memory over a longer time, and how long can this kind of memory be kept?
Exactly.
This would be really interesting, yes.
Yeah, I think it's nice. So, other questions?
Do you have any molecular markers for this drought-induced priming against disease?
No, we don't have them yet. We would like to identify them, but we don't have identified any yet.
Last questions?
It's my second question, but it is linked with my first question and the comment of the moderator. Do you investigate, do you know if stomata play a role during the infection process? Because this might explain something. Because if you first induce drought, you close stomata, and then less infection. And what you monitor is resistance. But when you concomitantly infect and induce drought, maybe pathogens, but I'm not familiar with the symptomatology of these pathogens. But this may explain a little bit of this.
So to answer that question, barley is only susceptible through the open flowers. So you need open flowers, and it's entering via those open flowers into the tissue. And the second point that I would like to highlight here is that we've performed the infection under 100% humidity. So in all of our samples, we've put a plastic bag around the heads in order to have really high humidity, so that all possible pores are open and to have an equal infection environment. So I would kind of exclude those facts.
Okay. Thank you very much, and please help me to thank you for the presentation. And again, apologies for those technical difficulties. Thank you very much for that.